This is episode 264 of the Stem Cell Podcast, Planarian Regeneration with Dr. Blair Benham-Pyle. Hey, everybody, we are Daylana and Arun. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the podcast, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Blair Benham-Pyle from Baylor College of Medicine. She's on the podcast to talk about her research on the tremendous regenerative potential of the Planarian flatworm. We've also got our usual roundup of recent highlights and stem cell news. That's coming right up.
But first, the world's best and brightest in stem cell research and regenerative medicine convene once a year for the ISSCR annual meeting, and you are invited. Join more than 4,000 scientists from around the globe at ISSCR 2024 to take part in a 4-day program comprising the year's most significant new advances in the field. Learn more at isscr2024.org.
All right, you guys, for the first roundup story today, there's a bit of overlap here. Dr. Benham-Pyle's at Baylor now, but she had some overlap with you at Stanford, Arun, and I've got a story from one of Stanford's most illustrious scientists. That's Dr. Irving Weissman. This is a story also with Kim Hassenkrug, who's at the NIH Laboratory of Persistent Viral Diseases. That's at the NIAID. And that relates kind of backhand to this story. It's a story about hematopoiesis, as you might expect. Irv, the godfather of hematopoiesis and all the CD molecules. And as we all know, I've talked about many, many times on the show, the HSC, my favorite cell, can generate all the cells in the blood, and it does. But there's some functional heterogeneity amongst HSCs that's emerged. We had this traditional paradigm that it was, the cell lineage hierarchy. A single HSC gives rise to all the cells, and the diagram would make you think that it's in equal measure. But there's a diversity in the degree to which hematopoietic stem cell contributes to lymphoid or myeloid cell lineages. And as you age, that kind of skews. There's at least 2 subtypes of hematopoietic stem cell that have been described. The one kind of mainline HSC is the one that, gives rise to all the cells in equal measure, balanced hematopoietic stem cells, as it's called, with equal production of lymphoid and myeloid derivatives. And then there's the more pathological hematopoietic stem cell that's emerged in recent years, and that's the myeloid biased hematopoietic stem cell. And as I alluded to, the proportion of those myeloid biased hematopoietic stem cells and the degree of bias increases with age, resulting in increased myelopoiesis, and by virtue of the fact that you have reduced lymphoid differentiation, you have reduced adaptive immunity. Also, you have what's called inflammaging, portmanteau there of the inflammatory results of the aging hematopoietic system, and also like real pathology, myelodysplastic syndrome, chief among the myeloid-related diseases and all derivatives thereof. So in this story, it's pretty straightforward. An approach that totally makes sense and is built on many of the technologies that have been developed in the Weissman lab and elsewhere, and that's this kind of depletion method. We've talked about it on the show before, using antibody-based ablation of the hematopoietic system in advance of bone marrow transplant, and this is a kind of related approach. And the idea is, can you use antibody-mediated depletion to reduce the degree of myeloid hematopoietic stem cell skew, right? By specifically targeting those myeloid-leaning hematopoietic stem cells. So, as I said, very straightforward using the approach that was developed already. They identified specific cell surface markers or a combination thereof of cell surface markers that would target these myeloid skewed hematopoietic stem cells. And those antibodies that they arrived at, a triple cocktail that targeted CD150, CD47, that we all know so well, as well as KIT. Um, and showed that, in fact, if you could, well, first showed that that cocktail could deplete these myeloid skewed HSCs, and then showed that if you uh, implemented this depletion in aged mice, you could endow them with a more youthful immune system. So increasing the degree of the lymphoid uh, progenitors, generally speaking, speaking to the more balanced differentiation after depletion of those myeloid skewed cells, including naive T cells and B cells, which will presumably increase adaptive immunity. Also, decreasing these age-related markers of immune decline, namely pro-inflammatory markers like IL-1 alpha, beta, CXCL2, and 5, and that's a key facet of this story, I thought, because the inflammatory sequelae of aging, aging hematopoietic system, and aging throughout your body and all tissues. That's a real. That's part of aging, right? And that's part of our decline. So the idea that you could reduce those pro-inflammatory markers secondary to this myelodepletion, I thought that was a pretty big deal here. And of course, yes, you improve the adaptive immune response to viral infection. And that's how I think it circles back to Dr. Hassenkrug's involvement in this story, is that this is a means of improving the response to viral infection, right? Which is a big deal and has emerged, um, you know, the degree to which that's a scourge in our society has really been underscored in recent years. So this is again, straightforward approach that I think has tremendous implications. Last thing, they finish by showing that they can target, there's some uh, for diversity in the marker expression between mouse and human hematopoietic systems. They show by targeting homologous markers in a human that they can also presumably they can target these myeloid skewed HSCs in a human hematopoietic system, setting the stage for potentially using this in a clinical context in humans and aged humans. And I mean, that's a huge deal. There's been countless stories talking about the rejuvenation idea, the whole Dracula with Amy Wagers and others. But here's a antibody based approach that's not so much about like the transfer of a hematopoietic system between young and old mice, as much as just targeting the bad cells to enrich for a more balanced hematopoietic system. So it's a big story. Of course, Irv is gonna wake up in the morning and write something down. They might publish it in a Nature journal, but this story merits that degree of attention, I think, because it's so clinical, and that combined with all the other technologies developed in the Weissman Lab and elsewhere makes me think that this type of approach could be really close at hand. Arun, what's your take?
Yeah, Irv gonna Irv, as they say. But yeah, this is just another hallmark study in the decades of amazing studies that have been coming from the Weissman lab at Stanford. And you're right, I think there's definitely a very strong interest these days in this concept of rejuvenation. We've heard of entire companies and startups being formed in this area, like Altos Labs, for example. Their whole idea is rejuvenation in a different context, using, let's say, the Yamanaka factors to turn back time and induce rejuvenation in that way and address aging. But there's other folks here at Stanford. And in fact, you alluded to some of the other folks working in this area of, transplanting the entire hematopoietic system from one animal to another. Parabiosis is that other example, right? Tony Weiss-Correa over at Stanford has been doing a lot of this, where you basically sew together the circulatory systems of two mice, and an old mouse and a new mouse, and you have sort of an aging reversal phenotype that happens there. Here, and I agree with you. I think I like this because it's a lot more targeted, so they can specifically target those, myeloid skewed HSCs causing the aging phenotypes here using this cocktail of just 3 antibodies. I think that's pretty amazing. I was also impressed that even though there's just 3 antibodies, 2 of those antibodies are somewhat generic, generalizable. You know what I mean? CD47, they've been talking about that a lot. Don't eat me signal, but KIT is also a pretty pan proliferative marker. So that's pretty impressive. You can really target it just based on those three antibodies. Like what you're alluding to at the very end there, the human side of this is what gets people really, really interested. Of course, you can demonstrate all these phenotypes in the mouse, but if you can show the initial proof of concept in a human system, then inevitably that's going to go towards clinical trials. And I'm sure that's exactly what they're aiming to do here at Stanford.
Yeah, I'm glad you brought that up. I mean, I brought it up at the end because, yeah, this is a translational story, which is why it's in Nature. But yeah, the three antibodies that are, I wouldn't say ubiquitous, but they're also on other hematopoietic cells, right? Not to mention other tissues throughout the body. So yeah, you wonder if in scaling this up to a human system, given the broad genetic diversity and like, the pathologies that we're talking about are also very diverse as opposed to like this very controlled experimental system with the aged mice. So we'll have to see how that plays from a safety standpoint. But as you said, I'm really impressed with how this approach has evolved from a more sci-fi, parabiosis kind of cartoonish application to breaking it down mechanistically. And other studies are looking at the factors like GDF, et cetera, that may be, the silver bullet. Um, but all these studies converge on kind of repairing an age system. And I think as the technologies continue to advance, I think we're going to arrive at something that's not only effective and safe, but practical, vis-a-vis parabiosis. I don't know if that's ever going to play out. So I'm very optimistic. And as you said perfectly, Irv is going to Irv. And as he continues to Irv, he's getting closer to the goal here, which may just have Irv continuing to Irv for, an indefinite period of time. I think would be a win for science.
I totally agree. I would love to let Irv do Irv things forever, ever and ever and ever. He's the best. He's totally great. So moving on to a cell stem cell story coming from Duke University. Go Blue Devils. This is a an interesting concept. It's basically connecting two portions of the body where you may or may not think they really have much connection. This is the gut brain, and there's sort of a gut brain axis that's starting to be fleshed out a little bit more here. There's actually certain connections between the gut and the brain in context of neurodevelopmental disorders and neurodegenerative disorders, like even Alzheimer's, for example, and some connections there, where say the microbiome could have an influence in the development of some of these disorders. This is talking a little earlier than that. This is looking at the subventricular zone in the brain and how stem cell niche injury in that part of the body could be associated with intestinal perforation, if you can believe it, in the gut in preterm infants. Okay, so the whole idea is, kids sometimes are born with these intestinal issues, right? Intestinal perforation, sometimes caused by necrotizing enterocolitis, and that can lead to neurocognitive disorders. So they wanted to flesh out that connection here and do that in a more mechanistic way using actually a mouse model, which is pretty wild because the whole concept of this story was that association, which has been detected in these infants and these babies who have been born with these problems. Right? They have intestinal perforation, and then it leads to downstream neurocognitive disorders, right? It's linked to lifelong neurologic impairment. Mechanisms are not super well understood. So, what you want to do is be able to diagnose these preterm brain injuries. And that's still a challenge in itself. So, here, they're actually able to identify a marker called subventricular zone echogenicity, which, and again, I'm not a neuroscientist, so this is a little bit of French to me, but I think it's basically like these little gaps that are found on the ultrasounds of the brains, if I can interpret it correctly, these cranial ultrasounds in preterm infants after they're born. And the association here is again, some of these kids who are born with these SVEs, subventricular zone echogenicities, are the same kids who have intestinal perforations at birth. So there's an association there, and they flesh that out through the clinical data set in humans. All right. And then confirm that down the road, it's significantly associated with motor impairment at 2 years. All right. That's just based on clinical data sets. And then the cool part of this is, you might think that association is wild enough, but then they managed to reproduce it in a mouse model, right? So they actually replicated that SVE, subventricular zone echogenicity phenotype by inducing intestinal perforation in these really young mice. And then, confirmed through echo and all these other examinations in the mice and the pathology that yes, those mice that had the intestinal perforations also had that same hallmark phenotype in their brains. And then I think what took us to the level of cell stem cell papers, of course, you got to do the mechanistic deep dive as to how this association is actually happening. All right. And they found that there's an NF kappa B signaling or inflammasome assembly phenotype here that's actually in these multiciliated FOXJ1 plus epididymal cells, which apparently there's some inflammatory regulation. We're talking about inflammation, right? So perhaps there's some sort of inflammatory phenotype that's going on here that could be inducing that gut brain cross interaction, right? And so what they're showing here is that there's a loss of the ependymal border in this postnatal stem cell niche induced by the intestinal perforation in the gut of these mice. So ultimately, these data are suggesting a mechanism for that preterm brain injury actually localized to the SVZ that hasn't really been described before. And in the human side of things, it's really determined by the ultrasound, and it was just the association. But in the mouse side, it was really fleshed out by this inflammatory signaling cascade. I think there's still a ton that they need to do here in terms of the mechanism, and I'm not doing it justice by any means. But just even my superficial observation of this paper as with a something as distant as the gut brain axis, I think you really, really need to flesh that a lot more as opposed to identifying an inflammatory regulated phenotype, right? So I think this is setting up for a lot of future work that's going to happen there at Duke to identify the true mechanisms of this. But the novelty in my mind is just the figuring out this connection exists in humans and that you can actually recapitulate it in a model in mice. So that in itself is, I think, really, really cool.
Agree 100%. And yeah, a lot to do, but they kicked open the door, right? And that's what's so impressive and really grabs your attention about this. Yeah, it's a clinical translational story, and I love that Cell Stem Cell has showcased this as a clinical translational stem cell story because it superficially may not jump out at you as, but yeah, when as soon as you read the paper, you see how where the links are, and also just the importance of this, right? I mean, as a parent, and I'm sure you can identify with this, it's troubling to me that, infant will, you know, born with some intestinal perforation. They are in the NICU for a while. They get discharged because everything looks good, neuroimaging looks good, and then a year later, you got these motor deficits. Like that's shocking, and I think very troubling. So making the link between two compartments that are so distal, key. Reproducing it in a mouse, super key. And as you said, now kind of a playground for trying to connect and interrogate this link to define what the mechanism is and to identify biomarkers. I think that's another a point that was made in the discussion that's key here is that how do you distinguish these neonates, infants that are at risk from those who aren't? When you discharge them from the NICU, how do you predict those that may have some kind of neural sequelae? Well, first thing they're going to do, I hope, in these models is identify biomarkers that allow for distinction and follow up in those specific patients. So this is a story for me as a parent that has real impact, both in just illuminating links that would never have occurred to me, but also providing a platform for digging deep into that to address unmet needs. So a great story and great review, Arun.
Yeah, no, power in the model system. I think there's so many diseases out there that just you'd be surprised they don't have great model systems to study them, and this is one of them. And I think this, like you're alluding to, gives hope for folks and for parents whose kids might be afflicted with something like this to really get to the bottom of what's going on here. And there are limitations, sure, and I don't want to harp on that, it is a model, and we've talked about models a billion times on the show and the limitations with every single model, right? But one thing they wanted to dive into a little bit more was the sex determinant, right? Is there a role for male versus female in terms of determining the severity of a phenotype like this? I'm sure this is setting up for, like I said, a cascade of studies that's coming from the one and only Duke University.
Duke could have done better in the NCAA tourney, but I still love my alma mater, as do you. 25th reunion coming up in a couple weeks, Arun. Watch out. Moving on, we're just glorifying the models and what an amazing thing it is that you could recapitulate this link between the gut and the brain and the mouse. I'm about to kind of undermine the glory of the mouse as a model, but not to take away from that story, just to illustrate that while the mouse is our entree to our own biology, for the most part, it is somewhat limited. And a lot of that limitation derives from here, same compartment. We're talking about the brain development. Um, in a mouse, it's very different, right? Human neocortex, there's a lot more neuronal and glial cells that are pretty much the basis of our higher cognitive function. And it's about the size of our brains, of course, relative to the rest of our body. Not just the size, but the surface area and the folding of the neocortex. And that anatomy and expansion of cells is the result of progenitor cells called basal radial glial cells, also known as outer radial glial cells. And these cells are really abundant in humans, but rare, scarce in mice. And in humans, they live in the outer subventricular zone, right? Uh, so there have been a lot of experiments, including live imaging experiments, that have shown that these cells, the way you arrive at this expanded neocortex in humans relative to other model animals, is this higher proliferative potential of these cells. And also the transcriptome of these basal radial glial cells in developing neocortex of the humans has been described. And interestingly, these analyses, transcriptomic analyses, have shown that cerebral organoids are actually a really good proxy for fetal tissue. Transcriptomically, the cells in cerebral organoids and their behavior is very similar to what you see in the fetal brain. But the methods that have been used, most notably looking at the fetal brain, they don't really allow you to track the self-fate decisions that a single progenitor makes, leading to the broad diversity present in the human neocortex. And understanding that's really critical to picking apart the difference between these many species, higher, lower, well, different. I don't want to assign any value. But also not just evolutionarily, but to understand how these cells behave in the context of disease, right? So let's talk about the possibilities in describing the way these basal radial glial cells could behave theoretically. And we're speaking specifically about kind of lineage hierarchy and cell division. So there's differentiation, right? And there's theoretically three major ways that you can imagine this going. There's either a symmetric expansion or proliferative, where two radial glial cells give rise to two radial glial daughters, so progenitors just amplifying. Then there's a symmetric self-consuming, right, where you have a cell that just gives rise to two differentiated daughters, so spends all that progenitor potential in one shot. Or the classic asymmetric differentiation, where you give rise to one progenitor and one differentiating daughter cell. Even more so, though, there's ways that these cells can differentiate. It can differentiate directly to a neuron, which is called a direct neurogenic division, or they can get there by way of an intermediate progenitor. And that's a key distinction, because in the mouse, where the basal radial glial cells are really scarce, if you can recognize them at all, you look at instead these apical radial glia, and the way that they differentiate is via an intermediate progenitor. Okay, so getting to the story, this is a Nature Cell Biology story from the laboratory of Alexandre Baffet, who's at the Curie Institute in Paris. Sorry for the mispronunciation if I didn't get it there. And what the group did there is they developed this semi-automated live slash fixed. So in live tissue as well as fixed tissue, a method to look at the progenitor cell hierarchy in these basal radioglial cells in fetal tissue, but also in cerebral organoids. And what they showed is that there's a lot of amplifying divisions, um, which are symmetric, right? So the two progenitors, progenitor giving rise to two progenitors, but there's also these direct neurogenic division. So self-consuming progenitor giving rise to two differentiated daughter cells, but directly by this direct neurogenic division as opposed to going by a intermediate progenitor. And as I said, that's key because it's very different from the apical radioglial cells that we look at in mouse, which are completely reliant on the intermediate progenitors to amplify neurogenesis. So right there off the bat, we have a kind of a cell that's really scarce in the mouse, highly prevalent in the human, and accounts for kind of the different capacity of the human brain. And it's getting there by way of a different differentiation and expansion process. Also, add a little mechanism, identified that notch activation is evident in the self-renewing daughter cells. And last thing is, again, they underscored and demonstrated how faithful the cerebral organoids were in their behavior and the behavior of these progenitor cells and their differentiation, how faithful it was to the fetal processes. So for me, there, it's a nice point for organoids being a really useful model and proxy for fetal tissue, which, as we know, can be tremendously scarce, and elevates organoids as a model for unpacking this process to understand some of these evo-devo questions, and also, more notably, in the context of pathology of basal radioglial progenitors in development, fetal development, and brain development, but also perhaps in the context of disease. So a nice story about the brain, fundamental about how these cells expand to account for our big and brilliant brains, Arun.
Absolutely. Yeah, another great Nature Cell Bio paper here. Shout out to Stelios Lefkopoulos, who's actually on the show not too long ago for senior editor over at Nature Cell Bio. They do really, really great stuff. I mean, they are, of course, not exclusively a stem cell journal, but they cover incredible stem cell stories and developmental stories too. This is just another one. And I like what you're alluding to. There's basically two arms of this study that got me excited. One was the methodology and the methods development, and, creating this semi-automated live fixed imaging method to actually map out the radioglial division, not just in the fetal tissue, but like what you're alluding to, the real power of this is that this can be expanded to the cerebral organoids. And the second arm of this that really got me excited was exactly that, the application of these cerebral organoids, because in my mind, that's one of the most powerful applications that maybe is a little bit underrated when it comes to using these organoids, is using them for evo-devo studies like evolutionary development, seeing how these processes are conserved across different mammalian species. Every single time I see a paper come out that's something like in this area, it gets me really excited because it's something that's out of my wheelhouse. I don't do it a ton, but it's opening up that black box of evolutionary differences between species in a way that's really been impossible to do because, like what you're saying, there's such a scarcity of fetal tissues and just a scarcity of brain tissues to do these studies with. So, I'm just hyped up about a study like this because it helps open that black box.
Yeah, and while we're talking about it, I think it also illustrates and underscores the value of fetal tissue, right? At the same time, showing how cerebral organoids can provide a really faithful proxy, but you need to start with the bona fides, right? And this is a story here in 2024. God knows, probably the data was gathered 2 years ago, the way the journals work nowadays. But nevertheless, there's still a really outstanding need for fetal tissue in research. And this is a healthy reminder, I think, of the fact that while we're moving towards models that can obviate that need, we still need a ground truth and that is provided by fetal tissue. So let's just get that out there and try and drum up support for that avenue of research that has been very controversial in recent years in particular.
Yeah, and understandably so. But I will say the ISSCR, as the overarching society of stem cell research in the world, has done a great job in specifically addressing that and re-emphasizing the importance of fetal tissues for research purposes. So I think there's a lot of folks who share your interest there. Moving on to the last story of the day, last story of the roundup. This is another cell stem cell story, which is kind of fun because it's talking about autofluorescence. And I think when we think about autofluorescence, we always use it in the context of like a dirty word in science, like a curse word almost, right? It's like, oh, damn, I have some autofluorescence in my immunofluorescence staining. Can't pick anything up. I don't know if this is specific. I don't know if it's like just random stuff that's being popped up in my IF here. I don't know if it's real what I'm seeing, my phenotype, right? But here they're actually showing autofluorescence can be a good thing, right? It can be a biomarker of neural stem cell activation state, what they call it here. This is coming from the lab of Darcy Moore over there in Wisconsin. Wisconsin, of course, one of the hubs for stem cell research in the entire world. So we're talking about neural stem cells in some capacity over the last few Roundup stories, and progenitor cells and all that. And they're of course critical for producing neurons. They have to exit quiescence to make neurons, right, and activate their cell cycle and all that. But actually, our understanding of that whole process is still restrained somewhat by the technical limitations of current technologies. We were just talking about imaging and how you might be able to use imaging to better understand some of these processes, right? And what they're using here is this thing called fluorescence lifetime imaging, or FLIM, of autofluorescent metabolic cofactors. Okay, so basically, this little puncta that they have identified in the NSCs, neural populations, neural stem cell populations, that shift in terms of their localization to lysosomes over the course of whether the adult mammalian neural stem cell is quiescent or activated. So the amount and the intensity of these punctate autofluorescence, or PAFs, shifts based on the state of that particular cell. And that's really the whole underlying concept of this story. So they found, using this non destructive live cell label free strategy, that quiescent NSCs and activated NSCs have unique autofluorescent profiles. This is really powerful because you don't have to do any sort of staining. You don't have to do anything to actually manipulate these cells. You can just look at them in their endogenous context, because that's what autofluorescence is, right? It's just naturally fluorescing stuff that's labeling things inside the cells. I actually deal with autofluorescence in a different context. I study doxorubicin, which is this drug that causes a lot of damage to the heart after chemo, right? Doxorubicin has a red autofluorescence, and it can be advantageous in certain ways if you're studying its internalization. Similar concept here. So they're using autofluorescence in a positive light. And what they found is that the quiescent NSCs actually display an enrichment of autofluorescence localizing to a subset of the lysosomes. And they can use that as a marker. Great, we're talking about biomarkers, right? So they can use that as a biomarker for quiescence and actually predict cell behavior at a single cell resolution. Really, really cool. And it's more than just showing through autofluorescence that, oh, these things are quiescent or not. They actually have to confirm that using transcriptomic analysis. So they came in with the single cell RNA sequencing to confirm exactly that these autofluorescent phenotypes are associated with their correlated transcriptomic profiles and phenotypes as well. So that's great. And ultimately, they're creating a catalog and also a method in some ways, an approach, what they call it, for tracking mouse NSC activation state, and ultimately expanding our understanding of adult neurogenesis. I think it's really exciting. And, to me, the next step for this is perhaps sort of obvious, is extending this concept of autofluorescence for quiescent versus activated stem cell state to other stem cell populations outside of the brain. How applicable could something like this be for all populations? I guess we'll find out.
Autofluorescence? It's funny because, like you said, I mean, it's bad. Yeah, I'm conditioned. Like, oh, autofluorescence, oh, yeah, we'll use that to gate out the bad cells. Great. Autofluorescence, that's my dump channel. Or like you're looking at an image and you're looking at it like, oh, what's that little spot right there? Oh, that's autofluorescence. Don't worry. We don't pay attention to that. So like we've been conditioned to ignore this result. And I think it takes like, you know, that's why you need people naive to science to be like, what's this? Because we're so conditioned to dismiss autofluorescence. And here it's a biomarker, right? So I would love to talk to the authors of this story and be like, what was the transition where you were like, yeah, 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 don't pay attention to that, and finally like, hey, you know what? Let's look at that because that's the toughest thing about science and entrenched thinking that I think hinders innovation. So I would love to know what spurred to reframe and recast that fundamental question. You know, a 5-year-old would look at a picture and say, hey, I see signal there, and it takes a postdoc or a jaded, investigator to say, we don't pay attention to that. So I love a story that says, no, let's pay attention to that and let's leverage it in a way that's really useful. I'm excited to see how this is deployed, not just in the neural space, but elsewhere, as you said. And also, I'm interested, I got to look deeper into this, into the dynamics, the on off of this. Can you use it to dynamically observe the activation state or quiescence of these things? Does it go back and forth? So many questions here. I mean, am I going to go look at my slides tomorrow and rethink all the autofluorescence? Probably not. But at least in the back of my mind, I'll have even more doubt of all my results, which is, just something that comes with becoming a more seasoned scientist, I guess. Doubt everything.
Hey, but that's part of being a scientist is you have to doubt everything. And even in science, where that is one of the fundamental principles of being a scientist is like be skeptical, right? We have certain things that we're so indoctrinated about, right? And this is one of them. When I was an undergrad at Duke, I remember this. I remember this very vividly. One of the first things I learned was how about autofluorescence and how you should watch out for that in your staining protocols. And that's, when I was in whatever year in undergrad. And ever since then, I've just established in my mind that this is a bad thing. All right. And so you're totally right. Shout out to these authors for deprogramming themselves in some way to take a kind of a clear look at this whole picture and say, hey, maybe this is not a bad thing. Maybe this is actually real. You're fighting against the norm, fighting against scientist psychology in some ways. You know what I mean?
Yeah, and why? On its face, it should be a good thing. Fluoresces in all channels. Isn't that good?
Yeah.
Well, we'll see. We'll see as the results churn out. And, while we're, going against the grain, let's just throw out a brand new model animal that we've never talked about on the show before in Planaria. But before we get there, I got a quick message from STEMCELL Technologies. Take your human pluripotent stem cell cultures further with M-Teaser Plus from STEMCELL Technologies, the most widely published medium for feeder-free human ES and iPS cell maintenance is now formulated for enhanced performance and versatility. M-Teaser Plus reduces medium acidosis for more stable cultures all weekend long. To learn more, visit www.stemcell.com/mteaserplus. All right, everybody. With us today from the Baylor College of Medicine, Dr. Blair Benham-Pyle, who is assistant professor in the Department of Molecular and Cellular Biology at the Stem Cells and Regenerative Medicine Center. Blair, thank you so much for joining us today on the show.
Thank you for inviting me. It's a pleasure to be here.
Well, I must say the pleasure is ours. We love having relatively young, new investigators on the show. And today, I mean, we've got a great one for you. I love that we were able to get you on this episode because you have a research focus that is at once so different from what we usually talk about on the show, but it's also pretty fundamentally aligned, I would argue, with our collective interests, right? We're talking about planaria, the famous flatworm. Tell us in your own words how you use the Planarian flatworm to interrogate stem cell biology and other important questions in biology.
Yeah, well, unlike us, planarians consist of many, many different cell types, but about 15 to 20% of their cells at any given time are highly multipotent adult pluripotent stem cells, which means that you can isolate these stem cells and you could inject a single stem cell into a lethally irradiated host. And about 25% of the time, that transplanted stem cell could rescue every single cell type in the organism. The other reason we study planarian flatworms is not just because they have a lot of stem cells, it's that they use these stem cells to achieve whole body regeneration on a very regular basis. So the planarian flatworm that I study, Schmidtea mediterranea, reproduces asexually, which means that when it gets big and is well fed, it sticks its tail down and it rips itself into multiple pieces. And each one of those pieces will regenerate every missing tissue and produce a clonal progeny that then goes on to repeat this process over and over and over again. And they never go through an embryonic stage of life, which means that these adult pluripotent stem cells are functionally maintaining this worm over multiple regenerative cycles. And so far, we haven't observed any decline in regenerative capacity over time. So yeah, we want to understand how those stem cells do what they do, how they maintain their potency, how they know what they're supposed to make, basically how they keep these worms alive forever.
Man, nature is just so cool, right? I mean, this is pluripotency to the max in an adult system, something that we don't always think about here on the show. It's just such an amazing and unique model system, the planaria flatworm. And as you've alluded to, one that's been so useful in understanding these fundamental developmental processes. And you're no stranger to dissecting some of those fundamental molecular mechanisms of development. I mean, if you've been doing this since grad school in some context, in some way, right? Grad school at Stanford, which is actually where we overlap for a little
Where
bit.
we overlapped, yes.
Exactly, small world. So when you were in the Nelson lab at Stanford, you had this really cool science paper focused on how mechanical strain induces YAP1 and beta-catenin activation to drive cell cycle entry. And as we know, YAP and beta-catenin and Wnt are such fundamentally important signaling pathways in development across a bunch of different species. So before we dive into the planaria side of things, take us back for a second to the study from grad school and maybe how it actually inspired you to continue in this area of understanding development at the fundamental level.
Yeah. So I came into graduate school with a background in biochemistry and biophysics. So I had been studying biological molecules from a physical perspective for a while. It was an intellectual interest, but I was fascinated by what the consequences of that might be for a cell. So if molecules can feel physical forces and alter their signaling behavior in response to those physical forces, what does that mean at a cellular or tissue level? And this was right around the time when we were starting to realize that stem cell fate could be regulated by their mechanical environment, and that stem cell differentiation can be influenced by the mechanical environment that you put a stem cell into. And I found that fascinating. And so I was curious what that meant in a multicellular context. So I wanted to understand, if you apply mechanical forces to a multicellular tissue, how do the cells respond? And we saw a really cool thing. So basically, the more force you put into a system, you kind of increase the percentage of cells that will re-enter the cell cycle, and the tissue can make these really subtle responses. But each individual cell was really only making a binary choice. Like, do I enter the cell cycle or do I not? And we could kind of connect that to the fundamental signaling mechanisms at a cell-cell adhesion complex through the actin cytoskeleton into the nucleus to activate transcription factors like YAP1 and beta-catenin, or co-activators. And I think, watching individual cells that are all clonal in a cell culture dish, they're all the same, but by increasing the stimulus, mechanical strain, I was seeing changes in probabilities of those cells doing particular things. And I think that was the first time where I started getting really interested in heterogeneous cellular systems and how a bunch of different cell types could collectively and cooperatively create really subtle and beautiful morphogenic processes.
Mm.
And so I started thinking about regeneration and growth in adult tissues at about that time. I knew that very cool stuff happens during development, but since I was in a cancer biology program, I was fundamentally interested in like what adult tissues do, how they age, how they avoid disease, how they can achieve wound healing and morphogenesis in a healthy way versus a disease inducing way. And so I started looking for regenerative models. I looked at zebrafish, I looked at sort of a bunch of different developmental systems. And then I started finding papers about these flatworms and these planarian flatworms, and you could cut them into a bunch of different pieces, and every single piece would regenerate robustly. It didn't matter where you took the piece from in their body, the cells would just kind of figure it out. They would reset their axial signaling gradients. They would induce proliferation and differentiation to create any missing tissue. It was basically, a bag of cells encapsulated in a body wall musculature. And they were all different, but they all were fueled and descendants of this adult pluripotent stem cell system. And it was just a beautiful self-organizing cellular system. And so as someone that hadn't come from a model organism, that didn't have a ton of genetics experience and was kind of stepping into organismal biology from a cell culture background, it was the perfect system for me. And so that's what drew me from sort of my tissues in a dish with mechanical stimuli into planarians.
Wow, the origin story. I can remember when that whole stiffness thing came in and the mechanical force driving fate, because it was such a bombshell of an idea. And now in retrospect, we're all like, oh yeah, obviously, because so many times in development, adult homeostasis, yeah, mechanical force has been implicated, piezos everywhere. But it's one of those things where the new concept comes in and then you got to look at everything over again. And it's funny to me, and the irony that you're working with Planarian, because it's such an old school model, right? I mean, that's like one of the first models that everyone as a scientist gets introduced to. But, your introduction to the model with the stiffness, you were in a really wide open frame of mind, looking at everything over again, and you're still doing it. I mean, that's the origin story, but I'll take you to the most recent thing that you got out. I caught this study on bioRxiv, and it's a similar idea. Here you're talking about spatial transcriptomics, right? Spatial came out, now the resolution's getting better, but now we got to take every question we ever had and throw spatial at it, just like we did with RNA-seq. And of course, yeah, lo and behold, we're getting really important new insights into everything. In your case, you're looking at this old school model with the new school tool. Elaborate on that study in the bioRxiv for us. Is there any homology? I mean, I'm guessing not, but maybe so, either in terms of like cell lineage hierarchy or function, anything between planarian stem cell niches and mammalian systems that we're more familiar with, or forget about, familiarity. That's what I want to latch onto to see how it informs my own system, but just blow our mind with what you've learned using this new tech on this old model.
Yeah. So, I mean, we went out like to find there's sort of a fundamental question in planarian biology, which is what makes a pluripotent stem cell a pluripotent stem cell, and what is it that allows these pluripotent stem cells to fuel whole body regeneration? Like that's the big question, right? And so for about, I don't know, over 10 years ish, I would say we've been trying to figure out if there are intrinsic properties to particular subtypes of stem cells within the planarian that can explain their remarkable regenerative capacity. And fundamentally, the stem cell compartment is very heterogeneous, but we haven't found the answer yet. And so one of the questions I had just finished a big single cell sequencing study of planarians and had found these transient cell states and a bunch of post-mitotic tissues all over the worm. that seem to be responding to injury. And I've always been interested in cell-cell communication and collective emergent properties. And so one of the questions I had was essentially, maybe it's not a stem cell intrinsic property. Maybe it's a collective property of a stem cell and its microenvironment. And we had been exploring gene expression across the animal in a bunch of different ways for a long time. But maybe the microenvironment doesn't actually change its gene expression all that much. And therefore, we couldn't find new markers of a microenvironment using differential gene expression. We needed to just figure out what's around stem cells. And so this sort of launched a really fantastic collaboration with another postdoctoral scientist in Alejandro Sanchez Alvarado's lab, Frederick Mann, who also got his PhD at Stanford around the time I did. And so he is coming from sort of a genetics, C. elegans background, and he's been really interested in developing tools and planarians. And he's always been sort of monitoring spatial transcriptomics as I had been. And he and I both really wanted to try this new method, slide-seq v2, in planarians to see if we could figure out how is gene expression spatially organized in the worm? And more importantly, can we use it in an unbiased fashion to just figure out what mRNAs and markers are enriched around stem cells in an intact animal versus a regenerating tissue? And so basically, this was our COVID project, ironically enough. Like, the Institute shut down right when we were planning this, but they allowed us in like sort of this relay team fashion handoff of samples to achieve slide-seq v2 during COVID. And so we cut worms, we embedded them in OCT, we handed them off to our histology core that sectioned them, and then handed them off to our genomics profiling core. And they prepped our libraries, and then they handed it off to our sequencing people, and they did the sequencing, and we got the data back. And we got to play with the data from there. And fundamentally, when you look at, so what slide-seq v2 does is it allows you to capture mRNA from a tissue section on a 10 micron diameter bead. And basically this bed of beads will capture your mRNA. And so any cells that are close to each other have a reasonably good probability of co-depositing mRNA onto the same bead. And so when you look at the mRNAs captured on these beads, it represents a small microenvironment of anywhere from 1 to 6 cells that are all in enough spatial proximity that they're co-depositing all of their mRNAs into this small space. And we could pull out all of the beads that had our stem cell marker PW1 on them and ask a very broad question, which is, what other mRNAs are likely to be co-deposited with a stem cell marker onto these beads? And the surprising answer is that it's a bunch of mRNAs from a cell lineage that no one studies, which are these secretory cells, which we've found a secretory cell lineage. It's sometimes called parenchymal cells, but I like to call them secretory cells because we now know by EM that they are packed full of these like vesicles, and they are clearly pumping out stuff in high volume. And when we looked at these secretory cells, there's actually close to a dozen different secretory cell types in planarians, but their gene expression profile is very stable. And no one has really studied them because their function doesn't seem to really change all that much in different injury contexts. They're just really good at pumping out what they pump out. But we found that stem cells are almost always closely associated with some type of secretory cell. And we focused on one in particular called, which we decided to call hecatonoblasts, because they're these like many-armed secretory cells. They're giant. And because they're secreting things, we think maybe there's some sort of fibroblast-like cell. We started out calling them like hecatonophages, but I don't think they're that type of cell. But anyway, fibroblasts fibroblast-like secretory cells, so we call them hecatonoblasts, and they express a ton of matrix remodeling enzymes. And actually, when we look at them, they're very closely associated with stem cells. And we decided to do an RNAi screen to knock down a bunch of genes that are expressed in these cell types. And we started to get some pretty subtle phenotypes, like slight decreases in proliferation, slightly smaller blastema. Fundamentally, I think they may not be required for regeneration, but they do seem to be closely associated with stem cells in the worm. We also found that stem cells are very closely associated with intestinal tissues, which we kind of already knew based on the animal morphology. And the stem cells sort of intercalate with the gut branches, and it had long been hypothesized and in some cases shown that the intestine provides nutrients that fuel stem cell proliferation. Actually, very similar to mammalian tissues where insulin signaling from the gut will trigger stem cell proliferation in nearby tissues. And so we also did an RNAi screen to knock down a bunch of genes expressed in the intestine, found really dramatic stem cell proliferation defects. And so I guess big picture, we were able to use this spatial transcriptomics approach to sort of, in an unbiased fashion, identify all sorts of tissues that are enriched near stem cells. And in the two cases where we investigated the cell type, we found some really fundamentally novel biology. One, if you disrupt intestinal patterning or intestinal signaling, you have dramatic effects on Planarian stem cell proliferation, which isn't necessarily a surprise, but we found it in an agnostic fashion. And then we got the big surprise, which is that we should probably be studying secretory cells a little bit. We investigated this one cell type, and it may not be important for regeneration, but we haven't ruled out its functions in other types of injury repair or tissue remodeling. And there are a bunch of stem cell microenvironments in this data set that haven't been explored yet, including associations with other types of secretory cells.
Yeah, it's a really amazing story, and it also kind of builds on some of the discussions that we've had recently here on the show about the importance of the stem cell niche, just in general, in regulating stem cell function and differentiation and so on. And I think the other part of the reason why this paper or this bioarchive manuscript is going to have a big splash down the road is because of the cool technology that you alluded to that was actually used to dissect the various niches in the flatworm. I mean, you mentioned the spatial transcriptomics, the slide-seq, which, I mean, it seems like we're talking about spatial on every single episode now, but also some really high resolution imaging that you used in your paper, which seems to be perfectly suited for these like translucent, semi-transparent model systems like zebrafish and like Planaria. Could you just reflect a little bit more about how the cool tech and how the cool toys these days are enabling you to make some of these fundamental observations in this old school model system?
Yeah, I had the pleasure to, I always have the pleasure of talking with lots of developmental biologists, and I consider stem cell biology a subset of developmental biology. But developmental biology is about observing systems of cells, perturbing those systems, and then getting to make cool, like, new discoveries about how those systems work. And the ability of scientists to observe and then perturb systems has dramatically changed over the last century. And so when I think about single cell sequencing and spatial sequencing and high resolution confocal imaging and single molecule fish and just everything we're able to do now, it allows us to observe systems in both a high resolution approach, but also an agnostic approach, which allows us to not have to decide what we want to look at in advance. We can look at every single mRNA in its spatial context if we want to. We can look at every single gene within its cellular context if we want to. And then we can understand and go back into the in vivo system to find those interesting things at high resolution in an animal during an interesting physiological process, and then target them, hopefully, and figure out what happens in the system when it doesn't have that component anymore. And so what I love about it is that it's great for discovery science. I mean, if you don't have to decide in advance, what you want to study or what molecule you want to investigate, it gives you permission to find molecules that no one knows are important yet, or cell types that no one knows are important yet. And so, yeah, I think it's an incredibly fantastic time to be studying developmental biology and stem cell biology, where we have all these tools that allow us to just explore agnostically.
Yeah, that's a key point. I'm glad you made it, because that's one of the, I guess, the pitfalls of academic science, all inquiry, is that you often find exactly what you're looking for, right? And when you open it up with these unbiased approaches, you see everything instead of looking for one thing. And yet somehow it always comes down to Wnt signaling. I don't know. Who knew? But seriously, I mean, looking into your work now, I mean, shifting gears a little bit, but staying on track also is, I relearned something that I know I learned before, but I promptly forgot. And that was what you mentioned, that planarians can reproduce asexually, but they can also reproduce sexually. I don't know if your particular strain does both, but yes, so some strains, maybe all, that can go sexually or asexually. And one track of your work, as you alluded to, it looks into identifying the environmental conditions or stimuli that promote or enable fission. So I got to ask, what's the aim there? And how do you expect the information gained to apply more broadly? And I want to like kind of drill down there, because I hate to do this. It's such a jerk move to try and say, oh, well, how does this apply to the high organisms? How you what's the clinical relevance? But you're writing grants, Blair. I mean, come on. So I'm sure you've already got the spiel right there canned for me. So if you had to sell this to someone like myself, who of a more, translational clinical bend, maybe, what would be the language to apply the insights that you gain from that asexual versus sexual environmental enablers, and how are you going to apply it?
Yeah. Okay. So, as you mentioned, planarians, especially Schmidtea mediterranea, exists in two biotypes. So Schmidtea mediterranea was found in a fountain in Barcelona, and we found two different strains. One is a diploid sexual strain that reproduces sexually. It's an obligate hermaphrodite. They form embryos. They lay those embryos into little cocoons, and then out hatches this highly regenerative little flatworm. But at some point in evolutionary history, the genome made a mistake. There's a chromosomal translocation from chromosome 1 to chromosome 3, and suddenly an asexual strain of Schmidtea mediterranea is born. We think that translocation prevented in some way germline maturation, and so a strain of Schmidtea mediterranea has been perpetually trapped in a juvenile adult state for thousands of years. We don't know exactly how long. The genome sequencing studies are ongoing. Jochen Rink is doing some of that work. But fundamentally, the question then becomes, the sexual worm doesn't asexually reproduce, as far as we can find under laboratory conditions. We've anecdotally maybe seen something like asexual reproduction or fission in the sexual worm under very rare conditions, but we can't reproduce it. And so why is it that the loss of germline specification led to this sort of increase in asexual reproduction behavior and this new emergence of a life cycle strategy that completely relies on regenerative capacity? So you have these two animals, one sexually reproduces, one asexually reproduces. So you can imagine, if you're trapped and you can't make offspring, there's lots of different ways an organism could survive. One would just be to live forever. Another would be to sort of rip itself up into pieces to grow the population. But then how do you decide when it's in your best interest as an organism to continue living or produce a clonal progeny that will go off and expand your line? And some might say it's stochastic, but at least in planarians, we know in our species of planarian, we've discovered that asexual reproduction is linked to growth and size. So why would an organism link a behavior like clonal reproduction to growth and size? Well, maybe there's actually an upper limit to how big a planarian can get healthily. Maybe there's a toxin that accumulates with time that needs to be cleansed, and regeneration is a way to do that. There's all sorts of questions around sort of growth control, size control, the limits of growth, maybe even the accumulation of damage that could influence how an organism makes that decision. Continue on as an adult or give up a piece of myself to the next generation of clones. And so I think it's really interesting to figure out how a genome makes that decision, how a system and a brain can sense the internal physiology of the animal and decide whether or not it's time to start again. And, for us, our genome starts again during embryogenesis, and there's lots of cool research about stem cell reprogramming and rejuvenation in the early embryo that's happening right now. And I think it would be very interesting if our asexual worms are, from an evolutionary perspective, doing something very similar during the process of regeneration. What if regeneration is a chance for this organism to start again from scratch, to reprogram the stem cell population to do something new, and maybe even at the end of that process have a much healthier and more competitive stem cell compartment?
Hmm.
I like that. It's like the concept of a clean reset, and I kind of wish we had that ability here as well as humans, right? Or higher vertebrates or whatever. And we've talked a lot about here on the show about how development and reproduction is so different between not just mouse and humans, but now we're talking about planaria, flatworms, and that's just a whole complete order of magnitude in difference in reproductive ability and the potential that these organisms can have in altering their reproductive trajectories. So it's really fun to think about. It's, stuff that we don't talk about a lot here on the show. So, I wanted to also shift gears a little bit because in addition to the amazing science that you're doing, that's just so incredibly unique, we also wanted to talk a little bit about your path in terms of becoming a new PI. And we like highlighting new PIs here on the show. I think we've been doing that quite a bit recently. You've actually taken a bit of a road less traveled among academic scientists. I think I can say that. You got your PhD in cancer bio at Stanford, like you alluded to, focusing on all things mechanic transduction. But then you decided to do a postdoc at the Stowers Institute for Medical Research. And in my mind, that's an incredible research institute, truly underrated in Kansas City, Missouri. But it's going against the norm in some ways because I think the old school traditional way of thinking is that if you're an American PhD, you have to do a postdoc on the coasts, so to speak, right? But you've turned that notion on its head and have had such an exceptionally rewarding and productive experience at the Stowers Institute. So could you talk a little bit about your postdoc experience there, working with Alejandro Sanchez Alvarado, and why you actually think the Stowers Institute is such an incredible place to be a biologist?
Yeah, absolutely. I'd love to. So I agree. When I told folks at Stanford that I was going to Kansas City, Missouri to do my postdoc, the next question was why? Why would you do that? And the answer is really the Stowers Institute. So the Stowers Institute for Medical Research is a unique philanthropic endeavor founded by Jim and Virginia Stowers. They gave an incredibly sizable endowment that allows, a dozen labs to be supported by hundreds of staff and millions of dollars a year in budget. And they provide dedicated research funding to support the scientists there and their staff. And so by going there, I was allowed to pursue really any question that I thought was the most important question that could be asked at the time, given the tools that exist in the community. And the Institute has a ton of very dedicated research staff across multiple cores whose whole purpose is to make science go faster, to onboard new technologies faster, It allowed me to onboard a new method for single cell sequencing, SplitSeq, that I used to generate one of the largest atlases of regeneration planarians. It allowed us to onboard SlideSeq v2 almost as soon as it was published, while it was still a preprint. And it gives you, it's an institute that really gives you permission to go after big questions, big important questions with new and emerging technologies, because you're a little bit less responsible for getting NIH funding. Now, obviously, if you get outside funding, it's great. Lots of the PIs do, they're very successful. But you have wiggle room to just pursue long-term science that's going to take a little bit longer. Maybe that'll take longer than the 5-year NIH trajectory, or that maybe just hasn't found its audience yet. You know, a lot of really great science happens before it has a big audience. And it's that cool discovery science that, you know, discovers Wnt signaling or hippo signaling or the next big developmental signaling pathway that's going to emerge, right? And so you have to have the room to find that new thing. And I think the Stowers Institute lets you do it. It honestly was the perfect place to be a postdoc, because I just got to focus on the research and the right questions and the right methods. And I mean, Alejandro too is a great mentor, and I got to learn from sort of one of the biggest influences on Planarian research in our generation. So it was perfect. Also, Kansas City is a great city. I'm just going to put it out there. Like, it's a good city to live in. Just ask Taylor Swift.
The champs. Yeah, I mean, not for nothing, but I feel like Planaria, and this is no shade on Planaria, because I think it's how it's one of those model systems that you really need to focus on to have critical fundamental breakthroughs, right? Cellular biology on a basic level, it needs to be so different from us that it's something that we're not looking for, so to speak. So I feel like a place like Stowers or any of these institutes where they have the infrastructure and the resources and also the courage to fund scientists that aren't doing, you know, the big headline grabbing research, that are doing the important research at the bottom of the pyramid that ultimately rises to the top, right? Wnt signaling, for example, everywhere. But yeah, love institutes like Stowers, the IMBA, in contrast to like Hughes or NICEF, for example. They're amazing institutes in their own right, but they fund science remotely. As well as having a campus, I know, with Hughes and Genelia, but I think it's more appreciated for funding investigators where they're at. I love the idea at Stowers and IMBA where they just create this playground and they just give you unlimited resources. And when I know it was at Baylor where you're at now, but it's a similar idea. When you were talking about your transcriptomic story, like you can only do a COVID study like that when you have all that infrastructure and all the partners where you're just handing the material handoff, you know, in a sort of like pass the baton type manner. So yeah, great spot. You know, and thank you for sharing the import of that type of work and that type of institute and everything else you talked about. I've had a really great time with this show, Arun. I'm sure I'm speaking for you too. We love talking about Planaria because it's just so much fun. And I've learned a lot here. Before we let you go, though, we're going to ask you a couple of peripheral questions. The first one being, if you were not a scientist, what would you be doing?
Weirdly enough, I think I'd probably be a lawyer or a law professor. So I was super into constitutional law and public policy as a high school student. One of the reasons I went to Yale as an undergrad is because I didn't know if I wanted to be a scientist or a lawyer. And so I took a ton of ethics and philosophy and law classes. And I even worked as a volunteer private investigator for the Office of the Public Defender after my freshman year of college. And what I figured out is I don't like criminal law, and I wasn't sure about corporate law. And I realized the only type of lawyer I really wanted to be was a law professor or an appeals court judge. And that's a long road. And so I always say, like, I made the decision sophomore year of college. I'm going to figure out what the truth actually is rather than arguing about what it should be.
Oh, that's so great. Because I was just thinking, the problem with the truth in law is that it's just not as satisfying as the truth in nature, because it's not actually the truth most of the time, isn't it? But great. I love that capsule there at the end. Finally, what is the best piece of advice that you've ever been given, either professional or not?
So the best advice that I think really changed my outlook on scientific training came from Madeline Julier. She's a professor of chemistry at the University of Pennsylvania. And I had the chance to meet her when I was still an undergrad and trying to decide what I was looking for in a PhD lab. And she told me that for my PhD, my job was to learn how to do science. And I shouldn't pick a lab based on the scientific impact or what kind of papers or what kind of prestige they had. I should pick a lab where I would be working for a scientist who was the type of scientist I wanted to become, because I was going to learn how to become a professional scientist. And I think that really influenced me. I think it helped me figure out that I wanted to be with someone who built a healthy research culture, who cared about like the professional interests and impacts that they would have both in academia and in the students that went on into industry or outside of science, a person that had a lot of integrity in science. And I think that was probably the best advice I ever got. Your training environment is going to pressure you to become a certain type of scientist. And every training environment you choose is going to push you to be the kind of scientist that it wants you to be. And so you should choose a training environment where they value the same things as you, where they want you to be the same type of person that you yourself want to become, because you're going to get pushed in that direction anyway.
Yeah, that's another great one. Because as you're saying, I'm realizing, and we've talked about it on the show, is that the tools change, right? And the knowledge base change, even the models change. Right? We talk about a new model every week on the show. But the approach, I think, if there's one thing in your control in charting out a pathway to success, which is so tough, right? And it seems arbitrary in many cases. And a lot of times it is about luck and circumstance. But I guess the one thing you can control is who you choose for your PI. So I think that's a really great piece of advice. And I just accidentally chose well in my life. If I had known what I was doing, I don't know that I could have done better. But a great piece of advice for all the young trainees out there. Find a leader that you'd like to be like. And Blair, I think you've found a lot of acolytes with your sharing today. You might be a few calls, a few emails in the inbox trying to join your lab. At least we hope so, because you're doing great stuff with the flatworm. My first experimental model. I might come back to it in a sabbatical sometime. Make some room, Blair. Thank you so much for sharing all your knowledge and insight, and really looking forward to having you on the show again. Hope you'll make it to the ISSCR. I mean, that is a stem cell model. If you do, we'll have a drink, throw back a can, and talk about the flatworm.
Sounds great.
Appreciate, again, you joining us. All right, everybody, that brings us to the end of our show. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on X at STEMCELL Podcast or via email at info@stemcellpodcast.com with feedback or to suggest guests. Thank you guys so much for listening. We'll be back at you in a couple weeks.